The oligo mass profiling, or OLIMP experiment, shown here, allows one to gain rapid insights into the structure of polymers in the extracellular matrix or cell wall of an organism. In the case here, cell walls are prepared from plant seedlings by first removing cellular components with 70% ethanol. The target wall polymer is next digested with a specific hydrolase, solubilizing particular wall components in the form of oligomers. An alternative approach is to digest the cell wall with the enzyme in situ directly on the tissue. The relative abundance of the released oligomers is determined by Maldi-Toff mass spectrometry and statistical analysis.
Hi, I'm Markus Pauly from the Energy Biosciences Institute here at the University of California in Berkeley.
Hi, I'm Markus Günl.
And I'm Sascha Gille, also from the Energy Biosciences Institute.
Organisms are surrounded by extracellular matrices that, depending on the organism, consist of complex polymer networks.
A rapid and sensitive way to assess the extracellular matrix is a method we call oligomer mass profiling, or short OLIMP, which we will show you today.
This procedure can also be performed directly on the plant tissue. So let's get started.
Begin this procedure by isolating the extracellular matrix or cell wall of plant tissues. Harvest 5 Arabidopsis seedlings, or the equivalent amount of other plant material, and transfer them into a 1.5 milliliter reaction tube. Make sure that the material is placed at the bottom of the tube. Add 2 3 mm metal balls to the reaction tube and snap freeze in liquid nitrogen. Then grind the frozen sample using a ball mill for 2.5 minutes at 25 Hz.